Good morning, good afternoon, and good evening, everyone. And welcome to the scientist.com and ProteoGenix webinar titled Advancing Your Innovation, Navigating the Complexity of Stable Cell Line Development. I'm Liam Sanyo from the events team here at scientist.com, and I'll be your host. Today, we're being joined by Dr. Laura Radu, Senior Account Manager for Recombinant Protein and Antibody Production at ProteoGenix, and Dr. Joe Verkaman, Senior Director of Business Development at Eurofins. Today, Dr. Radu and Dr. Verkaman will share strategies and solutions to streamline stable cell line development, ensuring high yields, clonal homogeneity, and seamless GMP transitions for biopharmaceutical success. And without any further ado, I'd like to welcome the first speaker, Dr. Laura Radu. Laura, thanks so much for joining us today, and the floor is yours whenever you're ready.
Okay, perfect. So thank you very much, Lion, for the nice introduction. So welcome everyone, and thank you for joining our webinar today. It is a pleasure for me today to present you this webinar on navigating the complexity of stable cell line development. Today, we'll focus on the vital role of stable cell line development in antibody production for biopharmaceutical development. CLD is essential for generating antibodies and recombinant proteins, ensuring consistent, scalable, and cost-effective production, which is critical for your project success. Selecting the right cell line and developing a robust CLD process lays the groundwork for future manufacturing and regulatory approvals. We will discuss the key steps in CLD, including gene insertion, screening, and selecting the high-producing clones, all of which are crucial for success. The goal is to establish a cell line that demonstrates high expression levels of the target protein while maintaining genetic stability and scalability. We'll cover optimization techniques and technologies involved, such as selection markers, automation in clone selection, and robust screening processes. Challenges are inherent in any CLD project, including low productivity, stability concerns, long timelines, and regulatory complexities. I'll share strategies to address common issues, troubleshoot productivity, maintain stability, and avoid development bottlenecks. With advances in bioprocessing technology, such as automated cell screening and high throughput analysis, overcoming these challenges is increasingly feasible. Finally, we'll explore how to advance your CLD project after initial development, focusing on transitioning from early stages to large-scale GMP manufacturing and preparing your cell line for transfer to a CMO for clinical and commercial production. I'll highlight the importance of collaborating with a CMO and a CRO that have expertise in CLD to accelerate timelines and minimize risks. So as we begin today's session, I'd like to introduce you to ProteoGenix, a leading CRO on biologics discovery and development. Our company was created in 1997, and in 2003, we established our commercial operation in Strasbourg, France. Today, we've grown to 160 employees, and we have over 21 years of experience in advancing biologics research. Our services cover the full spectrum of biologics discovery and development, including gene synthesis, peptide synthesis, polyclonal and monoclonal antibody development, protein production, and end product distribution. Now that you know about ProteoGenix, let's explore our comprehensive pipeline for antibody services, which offers an end-to-end solution from discovery to bioproduction. This seamless process ensures our customers don't need to coordinate multiple vendors to bring their therapeutic antibodies to life. So antigen design and production is the first step, which is followed by antibody generation, including polyclonal and monoclonal antibody production. After generation, antibody analysis follows. For sequencing needs, we offer hybridoma sequences and de novo antibody sequencing by mass spectrometry to provide complete data for further development. At the antibody engineering stage, we enhance therapeutic potential through affinity maturation, antibody humanization, AI-based developability improvement, and the creation of bispecific antibodies and antibody-to-drug conjugates. Finally, during pre-production, we ensure efficient, high-yield production with stable cell line development and transient mammalian expression. By partnering with ProteoGenix, you ensure speed, quality, and consistency throughout the entire antibody development process, saving time and reducing the risk of errors and delays. So the path to bringing biologics to market is complex and involves several critical stages, with stable cell line development playing a key role throughout. First, the process begins with in vivo and in vitro testing, where fundamental research and target validation help identify the right molecular targets for drug development. Once a candidate is selected, we enter the preclinical development, where CLD becomes foundational. This stage focuses on developing stable cell lines that produce the biologic compound consistently and meet regulatory standards for safety and efficacy. During the clinical development phase, from phases 1 to 3, the biologic undergoes human testing. CLD is essential here for preparing the investigational new drug submission to regulatory authorities, allowing clinical trials to start. After successful trials, we move to the regulatory review phase, where an agencies like AFDA assess the data to determine if the biologic can proceed to market. At this stage, the stability and scalability provided by stable cell line remain crucial. Finally, once approved, the biologic enters the post-marketing phase, which includes ongoing surveillance to ensure the continued safety and efficacy of in real-world use. In summary, CLD is fundamental to many of these stages, providing the foundation for reliable, scalable, and regulatory compliant biologics production. While navigating the complexity of stable cell line development, several key aspects need to be taken into consideration. The first one is the choice of cell line to be used. Selecting the right cell line is a critical step in stable cell line development, as it directly influences the production, scalability, and efficiency of biologics. Different cell lines have specific advantages and drawbacks depending on the nature of the therapeutic protein or antibody being produced. Key factors such post-translational modifications like glycosylation play a vital role in ensuring the biological functionality of proteins, particularly in biopharmaceutical application. CHO cells, which are Chinese hamster ovary, is one of the most widely used cell line in biologics production, especially for complex proteins. It has a well-established history for producing high-quality, scalable protein yields. CHO are also cells are often the preferred choice for large-scale production of monoclonal antibodies due to their adaptability and ability to perform human-like PTS. However, they may require non-extensive optimization to achieve the desired levels of yield and stability. While less common than CHO cells, HEK cells of are is HEK cell line is often used for specific types of biologics, including viral vectors. Its ease of genetic manipulation and human origin can be advantages um, for animal health applications, where cost effectiveness may overweight the need for complex modification. Other expression system can be used, such as insect cells. So insect cells offer flexibility for producing certain proteins, particularly those that require complex post-translational modifications. However, they may not always be the first choice due to differences in glycosylation patterns compared to mammalian cells. E. coli is sometimes favored over CHO cells, as they offer more affordable and faster production options with different optimization priorities. Ideal for producing simple protein, E. coli offers high yield and fast production. However, it lacks the post-translational modifications that may be needed for more complex therapeutics, such as glycosylation. So choosing the correct cell line is a balancing act between factors like scalability, production efficiency, and the biological properties required for the biologic. This decision is crucial as it will impact downstream development and manufacturing stage. Genetic instability is a common challenge in stable cell line development and can significantly impact the consistency and the quality of biologic production. These issues can arise due to several factors like gene silencing, unstable gene integration, disruption of regulatory elements, and accumulation of mutations. These issues manifest in several ways, like reduced protein quality, decreased cell viability, and very important, inconsistent expression levels. To address these issues, careful vector design and cell screening protocols are essential to ensure long-term stability and high productivity. Optimizing these elements early in the development process can mitigate many of these risks and ensure a more consistent and reliable production line. Achieving both high yield and long-term stability is often a trade-off in stable cell line development. Clones that produce high levels of recombinant protein may be more prone to instability due to metabolic stress or other cellular processes. Conversely, highly stable cell lines may produce lower yields, but their long-term performance is more reliable. Successfully managing the balance between stability and yield is crucial for the production of biologics, such as monoclonal antibodies, enzymes, and other therapeutic proteins. This balance can be achieved by carefully considering key factors like developability study, careful selection of clones that demonstrate both high productivity and genetic stability. Cell culture media composition also plays a critical role in the cell health, productivity, and stability. And also, consistent and precise control over the production parameters helps maintain the cell line stability. Any fluctuations in these parameters can lead to stress, instability, or reduced productivity. As we dive deeper into the development of stable cell lines, one of the most complex and crucial steps is the clone selection process that includes proof of monoclonality, high productivity, and stability from one batch to another, and of course, cost efficiency. In general, we can highlight two broad approaches to clone selection: traditional methods like limited dilution. These methods involve significant hands-on effort and are time-consuming and often introduce variability. While these techniques have been foundational in stable cell line development, they come with challenges in terms of efficiency and scalability. Recently, more advanced techniques have revolutionized this space. So, high throughput automation, image based technologies, and single cell cloning platforms like VIPs have greatly improved the precision and speed of clone selection. With this advancement, we can now achieve higher monoclonality assurance, more consistent productivity, and faster development timelines. The result is not just a reduction in the time to market, but also improve overall quality and reproducibility of the cell lines. As we continue discussing the complexity of stable cell line development, another critical aspect to consider is the substantial investment in both time and resources. The process of developing a stable cell line is complex and time consuming, often involving multiple phases of optimization and troubleshooting. For companies or researchers aiming to move their therapeutic or diagnostic products to market quickly, understanding and managing these resources is crucial. There are three key areas that heavily impact the efficiency and success of the processes. Using specialized equipment from bioreactors to high throughput screening platform and analytical tools, the equipment needed for stable cell line development must be highly specialized. Optimization that involves fine-tuning various parameters to ensure that the cell line exhibits the highest possible productivity and quality. And also expertise. So the expertise is involved the expertise involved is equally important. So navigating through complex challenges such as managing gene instability or low protein production requires a team of highly skilled scientists. In stable cell line development, regulatory compliance is not just a box to be checked. It is a critical pillar that ensures both quality and safety throughout the entire process. It is important to provide rigorous documentation at every stage of the development process to ensure product approval and the foundation for transitioning to GMP. This, as said before, this can be achieved by using advanced technologies that ensure accurate clonality verification and providing detailed reports that are essential for regulatory reviews. So before considering launching into a complex project like stable cell line development in-house, there are six key factors to evaluate. The expertise is the first one. Do you have the in-house expertise in stable cell line development? Is this something you already did before? This process requires a broad range of expertise, including genetic engineering, cell culturing in suspension and adherent cells, purification, using different methods like affinity, ion exchange, gel filtration, and characterization in multifactorial analysis. The second one would be the technology. You have the necessary state of the art platforms in place to develop and characterize your cell line. It requires extensive equipment like the lab setup, machines like VIPs, Biocores, HPLC, DSF, LSMS, to gather data and train personnel to perform and analyze the information and make informed decisions. The third one is if you already considered optimization of our sequence of interest before starting the stable cell line development project. Because anticipating developability problems right from the early stages of R&D is a significant asset for the proper development of your project. In addition, can you really afford the risk of failure? A cell line that expresses little to none of the biomolecule intended for bioproduction can jeopardize the viability of development project. It is well known that speed to market is a critical competitive advantage in biologics. How much time do you have to finish the project? Is it several weeks, several months, even years? Can you afford the delays when you are under short timelines for patent deposition, for example? And also, as your project progresses from research to full scale production, scalability becomes a crucial factor. Do you have the capability to scale up the production of your antibody of interest? When you consider these six factors, it becomes clear that the development of a stable cell line is a strategic step into the process of developing a new antibody, as it plays a crucial role in the final cost and quality of your biomolecule. Once you've made your assessment, if only one of these criteria is missing, then you need to start thinking about outsourcing your complex TLD project. This will allow you not only to benefit from specialized knowledge and resources, but you also reduce timelines and the risk of failure, helping you bring your biologic product to market faster and more efficiently. This is why partnering with a CRO that has all the previous criteria is the best solution, as it provides you with expertise. So CROs can bring significant expertise to the table. They have vast experience in developing stable cell lines for various therapeutic applications. This allows them to provide insights, tackle complex challenges, and make informed decisions that can streamline your project. Another major benefit of working with a CRO is the availability to provide guarantees. They can commit to deliver specific performance metrics on or timelines, helping you manage project risk and ensuring predictable outcomes. When it comes to timelines, partnering with a CRO can significantly accelerate your project. CROs have established and optimized workflows and dedicated teams that help shorten the time needed for stable cell line development, allowing you to meet important milestones faster. Faster, sorry. Developing stable cell line requires specialized equipment that is often costly to acquire and maintain in-house. CROs already have the state-of-the-art facilities, ensuring that you gain access to cutting-edge technologies without the capital investment. Finally, while it may seem counterintuitive, CRO partnerships can help you control costs. By outsourcing to a CRO, you avoid the substantial expense of building and maintaining the required expertise, infrastructure, and equipment in house. This allows you to focus your internal resources where they are needed the most. So, in conclusion, by choosing a CRO for your CLD projects, you gain access to a wealth of expertise, guaranteed outcomes, shorter timelines, advanced equipment, and controlled costs. Now, let's dive into ProteoGenix's streamline CLD workflow, which provides a clear pathway from the initial needs assessment all the way to scalable biologics production. This workflow is designed to ensure that at each critical stage, decisions are made with the highest level of scrutiny, guided by scientific rigor and data-driven assessment. A classic project begins with stable pool generation, where the primary focus is creating a pool of cells capable of reliably producing the biology. This is where we evaluate the cell and select the best performing pools, ensuring scalability and consistency in production. Step two involves single cell isolation, a critical phase where individual clones are selected for their ability to produce high yields of the biologic with the desired characteristics. Monoclonality is ensured using VIPS technology which precise and efficient clonal screening. Using VIPS reduces selection time by 50% compared to other methods like limited dilution, for example, and also ensures confident selection of top clones. Next step, we focus on cell line characterization to ensure stability and high quality biologic production. Here, we do an in-depth analysis of in analysis to assess key parameters such as productivity, stability, and overall performances. Finally, step 5 brings us to the final production ready stage where the clones are prepared for cell banking and scalable production. This is where the process is fine tuned and preparations are made for the transition to a full scale GMP production. So these are the usual steps of a classical CLD project. However, at ProteoGenix, we found that some of the most crucial steps occur even before stable cell line development begins, starting with a deep understanding of the customer's needs. We invest significant time in learning the project's background, objectives, and specific requirements, and this enables us to design and refine a customized roadmap that aligns perfectly with the desired goals. Additionally, we conduct an in silico manufacturability analysis of your sequence of interest to identify potential liability motifs such as glycosylation, oxidation, deamidation, or isomerization. A comprehensive report is provided detailing the identified risks along with optimized sequences to mitigate these issues, ensuring that an ideal lead sequence for use um, is for used in the next steps of stable cell line development. The proposed variants are transiently produced and characterized, and the results are discussed with the customer. And at this stage, a decision will be made regarding the choice of the lead candidate for the next steps. Throughout this workflow, we ensure that at each step there is a clear go no go decision point. So this allows us to move forward only when the data supports the decision, ensuring efficiency, scalability, and reliability of biologics production. Customers have the option to stop or pause the project at key points during the process in order to reassess the results obtained and next steps to be taken. So now I would like you to present the results we obtained for a recent project of stable cell line development. So a leading biotech company approached us to develop a stable cell line capable of consistently expressing a therapeutic antibody. The project's goal was to move forward from transient transfection to the generation of a robust, reliable cell bank, followed by characterization to ensure long-term antibody production. The project began directly with transient transfection to rapidly produce the target antibody and confirm its functionality, as the antibody sequence had been optimized previously during the affinity maturation process. So, to our surprise, the expression yield obtained was ridiculously low, around 6 milligrams per liter. However, the QC confirmed the high purity and integrity of the antibody. So, with the transient transfection results validated, we proceeded to create a stable cell line using our proprietary CHO-K1 cells with the objective of generating a higher expressing stable CHO cell lines that could consistently produce the antibody. So we use the classical approach, starting with gene integration, followed by selection and screening. So we screened 5 times 96 clones, so around 500 clones, to identify the highest antibody producing pools. So then the selected high producing pools were expanded and antibodies were characterized for integrity, stability, and aggregation. 3 high producing pools were identified with yields exceeding 2 grams per liter in fed batch cultures. At this point, the customer was thrilled with the improvement in the production yield and decided to continue the project with monoclonal selection by VIPs. So starting from the best expressing pool, monoclones were isolating using the VIPS technology in 96 well plates, and expression evaluation was performed by ELISA with anti-FC antibodies. We screened colonies seeded in 12 times 96 well, 12 96 well plates, and following monoclonality validation and growth assessment, 35 best performing clones were selected. The antibody expression was validated by SDS page and Alive. So the top 10 performing monoclones were expanded and cultured in fed batch conditions, and from these 10 monoclones, 4 of them showed expression yields between 3 to almost 5 grams per liter. So all clones demonstrated long-term stability, making them suitable candidates for scale-up. From the top 4 candidates, one of them was selected for scale-up and achieved excellent yields around more than 8 grams per liter, with optimized culture conditions. So we successfully developed a stable CHO cell line capable of expressing the client's therapeutic antibody with 100 times increased yield compared to the transient transfection. So in our pursuit of excellence in stable cell line development, it's crucial to recognize the key characteristics of a high quality stable cell line and how ProteoGenix leverages advanced techniques to guarantee success. So let's take a closer look. One of the first traits as high quality stable cell line is high productivity that is translated by long-term expression stability, where the cell line must consistently produce the target protein or antibody throughout multiple passages. At ProteoGenix, we develop monoclonal cell lines from selected pools, ensuring stability and high output through rigorous studies. The second essential trait is proofs of monoclonality. So using the VIPS system, we ensure automated monoclonal isolation through imaging. So VIPS tracks monoclonality, cell line survival, and growth, leading to selection of best stop candidate, which is a crucial step for downstream processes. Stable transgene integration is another essential trait of a high quality cell line. The gene of interest must be securely integrated into the genome to avoid loss or mutations. Our team uses stringent selection to ensure stable gene integration, preserving gene integrity and rigorous screening to remove aberrant clones, minimizing recombination risks, and ensuring consistent production. Final product quality is highly important as the developed cell line must produce a fully folded functional protein with optimal modifications and consistent production across batches to meet the quality standards of uniformity and efficacy. Our team conducts extensive assessment, including yield evaluation and SDS page under reduced and non-reduced conditions to confirm protein integrity. We also use advanced analytical techniques such as HPLC for purity, nano-DSF for stability, ELISA, SPR, BLE for antigen reactivity and binding kinetics, and mass spectrometry for protein integrity. In addition, using high quality expression and production media and monitoring across up to 90 generations will ensure consistent yields and stable quality. Last but not least, safety and regulatory compliance is required in order to ensure integrity and traceability of the cell line. So ProteoGenix performs stringent quality control of the RCB, taking into account pathogen testing like mycoplasma and bacterial contamination. We also provide complete traceability, so every operation, including batch numbers and COAs, are fully documented. In addition, regulatory-ready documentation is provided, including monoclonality report generated by VIPS with visual documentation ready for regulatory submission. So our robust approach to stable cell line development combines advanced technologies with stringent quality control, allowing us to meet the highest standards of productivity, stability, and safety. With ProteoGenix, you are assured of excellence at every step of the process. In addition, using our proprietary CHO-K1 cell line, customer retains the ownership of the cell line with no royalties, milestones, or additional fees during the later phases of the project. So once the stable cell line has been successfully developed, the next step is ensuring a seamless transition from development to manufacturing. This requires effective collaboration between the CRO that developed the stable cell line and the CMO. So the CRO focuses on optimizing the cell line for the desired antibody expression, while the CMO is responsible for scaling up the production process and ensuring regulatory compliance. In this process, we already have a strong and trusted partnership with Eurofins. Their expertise in manufacturing and their commitment to quality ensures that the characteristics and performance of the stable cell lines are preserved as it transitions to large-scale production. So this collaboration between ProteoGenix and Eurofins enables us to accelerate development timelines and deliver high-quality therapeutic antibodies efficiently. So my co-speaker, Joe, will share valuable insights into Eurofins' approach to manufacturing excellence. So with this presentation, I hope I convinced you that outsourcing such a complex project like stable cell line development is the best choice to reduce costs, timelines, and risk of failure, and why partnering with a CRO like ProteoGenix is a guarantee of excellence. So before I leave the floor to my co-speaker, Joe, I would like to inform you about our special offer consisting of a free audit and in silico analysis of your lead candidate before moving forward with the CLD production, with the CLD project, sorry. So thank you for your attention and don't hesitate to ask your questions in the chat.
Thank you for the introduction. Also from my side, I want to welcome everyone and thank you for joining our webinar today. It's a pleasure for me to add a few remarks from our side on how we see the interaction with ProteoGenix on the development of recombinant proteins. As we are focusing on an efficient way of producing these molecules, it's always beneficial to combine CDMO activities during the development of the cell line. In this way, we can have shorter timelines for the complete development, and by interacting with the cell line developer, we can also overcome some challenges and learn from potential issues during our further development. So let us start with a short overview of the activities that we cover within Eurofins, as well as how the CDMO part is incorporated. Within Eurofins, we can cover the entire scope of biopharmaceutical services, and this has been divided into several pillars, as you can see on this slide. If you go from the left to the right, you will see a part in discovery, providing in vivo models to evaluate the pharmaceutical effect of drug candidates. The genomics group is more focused on gene synthesis and sequencing, and the preclinical early development entity is doing biomarker and toxicological studies. At the clinical entities, we do analyze results from clinical trials, and our biopharma product testing is the most known, as it is a very huge network of testing facilities for pharmaceutical products. The final entity within this network is the CDMO network, on which I will elaborate a bit more and discuss the interaction with ProteoGenix on cell line development. I quickly jump now to our CDMO entity based in Ghent, Belgium, where we perform our development and manufacturing activities for biologics as well as chemical entities. On this slide, you can see the offices where we are located, and on top, you see the building with our manufacturing suites, and below are the offices for the development part. What is important to know is that we are at the same location, which means that we have a good integration and can save time on transfer of methods or protocols. We integrated the analytical activities, but also the formulation activities. As a third point, it's also important to say that all the people working with us have a can-do mentality and are showing a lot of flexibility solving issues during early development. Ghent is also a biotech hub with a lot of highly educated people, and at different levels you will find people with a sound scientific background to contribute to these projects. Within the facility, we can produce for human and animal health and have a focus on early clinical phases, which will go up to phase 2. When it comes now to cell banking, which is also the topic of today, we would be able to continue from the work from ProteoGenix and generate the master cell banks. But at a later stage, we'd also make the working cell banks and the end of production cell banks. The media optimization is also already being performed for us, but also here, this would be serum-free and animal component-free medium. The cell banks will be produced in a biosafety cabinet and then stored at 2 different locations. The cell bank characterization will be performed under GMP conditions, and release biosafety and stability testing will be done against all existing standards. On this slide now, we can see the integration of cell line development with CDMO activities in the early stages, and it shows why it's important to onboard very early the CDMO activities. When both activities are performed in a sequential way, it will take several months longer to have your first material ready. Next to this, some CMC parameters can already be assessed at an early stage. In our CDMO, we see clearly the benefits of accessing early clones and growing them at a small scale in order to start up the DSP development and generate material for further formulation development. This could also lead to selection of other clones and at least have a good idea of the performance of a clone within 2 separate companies. When the cell banking is being performed, we can continue with the different steps on product development, going to USP and DSP development, and a complete set for GMP manufacturing. Again, here, we integrated all these steps on site and have a complete CMC support available, as well as the formulation activity. We find it quite important that those activities are integrated, as our focus also lies on the development part for drug development. This slide gives you also an idea of the volumes we are using during development and GMP manufacturing for eukaryotic expression systems. As you can see here, we have a broad range of possibilities going from a shake flask to a small 1-liter bioreactor, but also up to a 200-liter single-use bioreactor. Although this volume is quite limited, we believe that achieving higher production levels during cell line development is important, as it will reduce costs at this stage. At the DSP side, we also like a challenge, and next to purifying difficult proteins, we do like to integrate formulation activities early on. For the formulation activities, we use, for instance, an all-in-one platform as the Uncle from Unchained Labs. This allows to screen multiple formulation buffers under various conditions. This will help to develop a liquid protein formulation, but also freeze-dried products, for instance. Next to this, we have our in-house platforms to screen even more extensive in order to ensure the best formulation for your protein. A side note on this is, for instance, solidification of biologics. We already had multiple projects going through a solidification step by spray drying the material and forming tablets. Material inside capsules as well as multi-particulate systems. This is really using the power of combining the knowledge of a pharmaceutical development entity together with the biological expertise that has led us to such interesting projects. A case study on this is a project that we have on colon targeting. These products could be used, for instance, for Crohn's disease. In this instance, we manufactured products that needed to be delivered to the colon, and a multi-particulate system was developed. This formulation also protected the materials within the colon, allowing to reduce the amount of material needed and having a better local release. So in conclusion, I wanted to mention that when the specific expertise of cell line development can be combined with CDMO activities, it has a big advantage in a project. These companies have a focus on innovation and try to excel in specific services. When these two are combined in an early stage, it will also allow you to save time in your development process. And as a last point, I want to point out that development at an early stage is key for the next stages within a project. When all the focus goes to large-scale manufacturing, some aspects might be overlooked, such as formulation development, and this can cause the early stop of a project. This concludes already my part of the presentation, and I would like to thank you for watching this webinar. Within this last slide, you can also find some useful information and people that can be contacted if you would need more information on our activities or if you want to discuss your project with us. Thank you.
Fantastic. Well, thank you, Dr. Verkamen and Dr. Radu, for the great presentations. And with that, we'll move right on to the Q&A session. And as a reminder, if you have a question that you'd like to ask Laura or Joe, you can submit it using the Ask a Question tab on the right-hand side of your screen. We'll cover all questions for Laura first, and then we'll move on to questions for Joe. Okay, so Laura, first question here. Which step in the antibody services process do customers find the hardest, and how does ProteoGenix help with that?
So that's an excellent question, indeed. So the hardest step is basically having the perfect antibody or protein sequence that will be um, used to move forward with the stable cell line development project. Because once you started this process, it will be very difficult to move to go back to the beginning. It will be time consuming and quite expensive. So we advise all our customers to make sure that the sequence of their lead candidate is basically a perfect sequence. And if it's not, we can, as I said before during the presentation, we can help them with that. So we can do an AI analysis, a developability and manufacturability analysis of the sequence, and we can advise them how to improve the sequence if necessary.
Excellent. Yeah. Okay. Next question here. So, at what stage of the antibody services do clients need to work the most closely with you, and how do you make that easier for them?
So working closely with customers is it's essential at each and every step of the process, basically from the beginning. It's a little bit related to the previous question. So from the beginning, analysis and ensuring that we have the perfect sequence of the perfect sequence candidate throughout all each individual steps of the project, and how ProteoGenix deals with that. So we have dedicated account managers, PhD and that are account managers that will follow up the project from the beginning to the end, and they are very present, and then they can update customers every 2 3 weeks and answer the questions. So there will be no black boxes and no you know problems with communication and everything. So that's
Fantastic. Yeah. Thanks a lot. Good question here. How detailed does the stable cell line documentation need to be? Do you just describe the main steps, or do you include things like chemicals and lot numbers?
we include as much information as possible. So we detail all the steps. We include details on the material, on the media, on the feeds, on the viability, on the growth of the cells. And so everything that all the data that is going to be issued during the stable cell line process is going to be delivered to the customer.
Perfect. A bit of a broader question here. What kind of project results can clients expect when working with ProteoGenix?
So, as I said before, we guarantee the expression yield. So, either at stable pool step or even at the beginning at the stable monoclonal steps. So, I think this is very important. It's a very important part that will the customers are looking at, and also that our GM stable cell line is royalty free.
Great answer. All right. Well, maybe you could just clarify for a minute how the developability assessment works.
Mm. So we use AI tools. So basically, that were developed in house. So they are proprietary tools that will look for protonational modifications like glycosylations, deamidation, isomerization, oxidation, but also hydrophobic patches and all other biophysical characteristics that could be problematic during stable cell line development, but most importantly important during manufacturing, because this is basically the most important step. This is a very important step during the process.
Yes, absolutely. Thanks a lot. All right. Next question here is your vector IP free?
Yes, the vector is IP free.
Okay, perfect. Maybe one last question for you then. How long does it usually take to develop a stable cell line?
So the shortest timeline would be 12 weeks from starting from the gene optimization and synthesis, vector cloning, stable pool development, and then monoclonal selection. Again, we have go no goes between each steps, and the customers are have the options to intensively characterize the pools and the samples, the antibodies that were produced by the pools, and of course the monoclones. So this will add couple of weeks in between the steps, but the quickest we've done is 12 weeks.
Perfect. Well, Laura, thanks so much for your time. And with that, we'll bring Joe back on here. Okay, there we go. Joe, all set to answer a few questions here?
Yeah, what makes them?
Awesome. Great. Okay, what would be the advantage of having a dedicated cell line development company next to your CDMO activities?
Thank you for this question. And I understand that most people are used to having all activities at one partner. At some point, we also considered of having cell line development within our CDMO scope, but we decided to keep this separate, and we do rely on other companies that are specialized in this field. This has more advantages for the companies we work with, as we don't force them to use a certain platform to develop their products. And next to this, we also believe that companies like ProteoGenix can bring some expertise to the table. So I do believe that a dedicated sign development company has an advantage, but an integration with other services as a CDMO is also important as well.
Okay, great answer. I think in the interest of time, we'll just make this next question the last one. Why would you perform formulation development during early development?
Very good question, and I'm happy that this question is being asked. I have also some background in formulation development myself, so it's an interesting topic. For what I see in a number of projects is that in many cases, formulation development is postponed to a later stage. However, quite often we see that issues with stability or bioavailability pop up during clinical studies, and then we have to accelerate formulation development to save a project. I can say that formulation development is important and it can help to differentiate from other products in a rather easy way. So, if you would ask me, I would definitely integrate formulation development early on, as it will save a lot of time at a later stage in the project.
Great answers. All right. Well, thanks so much, Joe and Laura, for your insights today. It's really been a pleasure having you with us.
Thank you, Liam, and also thank you, ProteoGenix, for inviting Eurofins for this webinar.
Thank you very much for having us, and we're looking forward to receiving your questions. Thank you.
Yep. Our pleasure. All right. A big thanks also to the audience for participating. And in closing, we hope you enjoyed this scientist.com and ProteoGenix webinar, and we'll see you again next time. Have a great day, everyone.